Assalamualaikum warahmatullahi wabarakatuh. My name is N.Izzati binti Mohzani, 1171483. Today I will present to you my paper review with title Duckweed'S Morphology and its interaction with microbiome. For introduction, duckweed is one of the unique plants that can be used as water treatment, has potential to utilize nitrogen, phosphorus, inorganic nutrient, and can be used to reduce the eutrophication. Duckweed also can be used as source of food since it contains vitamin, mineral, omega-3 fatty acid, unsaturated fatty acid, dietary fiber, and high protein value. For the water treatment, duckweed cannot play as the main role since it lacks of extensive root system, but it actually benefits for microbiome since it can provide surface area for the microbe to be attached. The interaction between the duckweed plant and the microbe that happen are benefit for both parties since the microbe can get the organic carbon and oxygen supply, while the plant can get the defensive immunity and the mineral as exchange. The objective of this paper review is to review the study about the history and morphology about the Lemnaceae family and its interaction with the microbiome that grow together in the growth medium. Then we move to the methodology. To achieve the objective of this paper review, there are 4 steps that has been followed, which is searching, screening, filter, organize, and report. For searching, the article, journal, and research paper has been searched out throughout the ResearchGate, NCBI, Academia, FAO, Google Scholar, and Science Direct. The keywords that has been used were duckweed, Lemnaceae, and interaction with the microbiome. For screening, it has been done by looking the title, abstract, method, discussion, and result to identify if it fulfills the inclusion criteria needed in reviewing this topic. For the third step, which is filter, the article, journal, and paper has been filtered and the exclusion criteria will be removed. This has been done manually by opening all the article, journal, and paper one by one. For the last step, which is organize and report, this paper review has been arranged according to the history and morphology of the duckweed, its growth condition, the location to be found, and the interaction with the microbiome. The bibliography also has been arranged according to the name of the author. For the quantity, 85 literature study has been chosen, but only 56 of it were identified relevant to the inclusion criteria. For the rest of it will be removed. For those literature study, 13 of it will touch about the interaction with microbiome, while the rest of it will touch about the history of duckweed, morphology of the duckweed, growth condition, and location to be found. Move to the first subtopic, which is history of the duckweed species and their morphology. For the history, duckweed plant early discovered in middle 1950s by Elias Landau from ETH Zurich, Switzerland. For duckweed family's name, there was a confusion happened whether to put Lemnaceae or Araceae due to 140 million years ago, the taxonomy position in duckweed species started to be split, and there was a species from Lemnaceae family, which is Spirodela polyerhiza, has similar characteristic with a species in Araceae family, Piscia species. Due to that, the argue about the family name started. Lemnaceae family were divided into 2 subfamily, which were Lemnoideae and Wolfioideae. Under subfamily Lemnoideae contain genera Lemna, Spirodela, and Nondoltia, while subfamily Wolfioideae contain genera Wolfia and Wolfiella. There are 7 genera has been recorded, which are Lemna, Spirodela, Wolfia, Wolfiella, Wolfiopsis, Pseudo-Wolfia, and Landoltia, but Wolfiopsis and Pseudo-Wolfia, both species under this genera, were synonymous with species under genus Wolfiella. Duckweed species under each genera is stated below in table. As we can see, Lemna contain higher number of species, which were 13, followed by Wolfia 11, Wolfiella 7, Spirodela 5, and Landoltia 1. Next, we move to the second subtopic, which is duckweed growth condition. Duckweed species can be found worldwide and in wide range of temperature, growth nutrient, and pH. For nutrient, duckweed can adapt as low as 1 to 3 degrees Celsius and up to 35 to 40 degrees Celsius. The duckweed can grow optimum at 20 to 30 degrees Celsius. For nutrient, duckweed can adapt with nitrogen, phosphorus, potassium, calcium, magnesium, sodium, bicarbonate, chlorine, and sulfur. For pH value, duckweed can adapt with pH 3 to 10 and optimum growth at pH 6.5 to 7.5. Now we will touch about the third subtopic, which is location. For location, firstly, we see the location based on continent around the world. Duckweed can be found in North America, South America, Africa, Europe, Asia, and Oceania. Based on the map previously, Asia is one of the continents that can be found with duckweed. Research has been done and it found that duckweed also can be grown in Malaysia. As we can see, this map shows the state that duckweed has been discovered by the researcher. The last but not least is interaction with microbiome. Microbe that can be found in aquatic plant is similar with microbe that utilized by terrestrial plant. Symbiotic interaction between duckweed and microbe can either be mutualism or commensalism, and it also depends on the adaptability of both species with the surrounding. Researchers discovered that duckweed also can associate with microbes from phylum Proteobacteria and Comammodaceae that naturally exist in wastewater. Duckweed also can interact with microbes from other plants, which is microbes from Japanese leucanthrife plant. Apart from that, duckweed also can cope with additional microbes that not from wastewater, such as Pseudomonas species, Vibrio species, Klebsiella species, Enterobacter species, and Serratia species. Aquatalia magnusoni strain H3 microbe also can improve growth of duckweed up to 11.7 until 32.1%. These are all the references that has been used. That's all from me. Thank you.